Epithelium and Junctions. Here we see a host of different types of epithelial cells. In the esophagus, we had stratified squamous epithelium. That's also what we have here in skin. Stratified squamous epithelium is non-keratinized in the case of the esophagus, but we do have it there. Trachea, we have ciliated cells, pseudostratified columnar ciliated cells. In the stomach and the intestine, we have simple columnar cells. In the urinary bladder, we have transitional epithelium. And so in different parts of the body, you have different components. Another type of epithelium is that associated with glands, glandular secretions. And here we can see the different types of epithelium. Stratified squamous, in other words, it's flattened on the surface, and it's more than one layer. Multiple layers flattened on the surface is stratified squamous. Sometimes it's really highly keratinized on the surface, as you see there, like maybe the heel of your foot. Here we have pseudostratified columnar epithelium, which has ciliated cells on it associated with the respiratory system. Transitional epithelium has scallop-type nuclei on the surface for the urinary system. Also, we have simple columnar epithelium that we have lining the gut. And if we look at one of those cells of the gut, and this is in the basal region. This is collagen right in through there. There's a basal lamina at the bottom part of the cell, and so we're really at the bottom part of these tall simple columnar epithelial cells. You see a host of mitochondria, lots of mitochondria that we see in through there. Interdigitation between adjacent cells, so this is one cell, another cell, another cell, another cell. This is the nucleus with the euchromatin, heterochromatin, as we see here. That one has a nucleolus, a euchromatin, heterochromatin. These two cells here are lymphocytes migrating through the epithelium, but you can see a host of mitochondria at the bottom associated with these cells. Again, we can see another one. Here we can see the basal lamina going between two adjacent cells at the bottom of these intestinal absorptive cells. Again, we're at the bottom of these intestinal absorptive cells, and also you can see other cells in here. This is a plasma cell, a lymphocyte. This is a blood, this capillary at a pericyte, another lymphocyte in through there, and a macrophage. So we have a host of connective tissue cells adjacent to the epithelial cells, and we see blood cells, blood vessels that provide the nourishment for the epithelial cells, which are non-vascular. In the kidney, we can see the PAS staining in the thick membrane, basement membrane within the glomerulus itself. Also, we can see the PAS staining of the brush border of the proximal tubule. In addition to the apex, you also see the base. The base is also PAS positive because that's the basement membrane. Very characteristic of epithelial cells is to sit on a basement membrane. We can see that again, the basement membrane is right in through there, connective tissue cells here below. These are simple columnar epithelium. You can see the brush border, which is PAS positive, and goblet cells as well. The secretions of the goblet cell are also PAS positive. If we look in the trachea, we see pseudostratified columnar epithelium. They're columnar in shape, they're ciliated cells. It looks as if it's stratified, but it's really not stratified. All the cells touch the base. They don't all touch the surface, but they all touch the base, hence its name, pseudostratified. And this is what we see here, the ciliated cells, and sometimes we have a goblet cell. So this is the airway of the trachea, cartilage over through here, and we can see other nuclei in those fibroblasts in here, but this is epithelial cells that line this space. Again, we can see those epithelial cells in the trachea right in through there, and also we see glands, and glands themselves which put moisture in the airway, those too are epithelial secretory cells. Here another one, we can see this is a false vocal cord, true vocal cords in through there, but most of this is actually this is a view right in through there. This is a false vocal cord, stratified squamous epithelium, as opposed to ciliated epithelium. Here we can see the ciliated epithelium a little better, see the cilia and the basal bodies in through there. So this is pseudostratified columnar epithelium, just like we've been describing with ciliated cells on the surface and some goblet cells as well. Now, the epithelium is specialized, and it maintains that polarity where it has a base and it has an apex by junctions. And we can see the different types of junctions. You have the zona occludens, which is the one at the very top and through here, zone, so it makes a belt around it, and it prevents things from going through, hence its name, occludens. That's a tight junction. The zona adherens interacts with actin filaments to help hold the zona occludens open. A zona adherens is a belt that goes around there that has actin filaments that interact with it to support the zona occludens. So you have zona occludens, zona adherens, and then desmosomes. The desmosomal connections that we see in through there, these are desmosomal connections, spot wells to hold cells together. The other junction is the gap junction, and here we see the gap junction in through there, and that provides communication from one cell to another cell. So desmosomal connections are throughout here, which are spot wells to hold the cells together, as opposed to zona adherens, which forms a belt that goes around. And here we can see those again, zona occludens, which occludes the things from going through, zona adherens, which has the actin filaments interact with these guys, and then you have the desmosome, which are the spot wells, and they actually interact with intermediate filaments. So you see the zona occludens, zona adherens, and desmosomes in through there. If you look at those at the light microscopic level, you can see these kind of bands or bars, so-called terminal bars. And the terminal bars are these three junctions, the zona occludens, zona adherens, and the desmosomes. We can see those here on this electron micrograph 6B, zona occludens, zona adherens, and these are different desmosomal spot well connections that we see there. Again, zona occludens, zona adherens, and the desmosomal connection. Zona adherens again, zona occludens, and desmosomal connections that we see over there. Here again, we can see the similar type of junctions. Here's a desmosome connections, zona adherens, zona occludens right in through there. We can't really tell which one's which, but both of them are there. Zona occludens at the very first one, adherens is the second one in line. Even between the goblet cell and the intestinal absorptive cells, we would have zona occludens and zona adherens in through there, even though we can't really see the detail associated with it at this magnification. Now, the zona occludens prevents things from going through because there is a welding of the two cells together. And here we see one cell there and another cell here, and we see this network-like connectivity between the two. And that brings us to the E and P phase. The E phase is the ectoplasmic, or the outside. The P phase, or the particle phase, is a part of the leaflet of the membrane that projects to the inside. So if we see that this is on the inside of a cell, that's outside of a cell, if you split it through there, you would see the ectoplasmic phase and the protoplasmic phase, or the P phase. So here again we can see that the ectoplasmic is the outside part, and the protoplasmic which you think about particles of protoplasmic because there's more particles on the inside. And these are integral proteins on the inside than they are on the outside. So if we look at this with a higher magnification, we can see actually two cells. This is the ectoplasmic face and the P protoplasmic face with all the particles in through there. If you look in through there, it looks like little ridges. In contrast, if you look over here, it looks like indentations, because this is the outer leaflet of the cell that got removed here, and this is the inner leaflet of the adjacent cell. So ectoplasmic, protoplasmic faces that we're seeing there. This slide shows you a carbon replica transmission electron microscopy of the gap junction, and you can see a series of little channels going through there, which is the junctions themselves, little channels that are going through there, and you have proteins lined up around each of these with a little hole in the center with things being able to be transmitted from one cell to another cell. In the liver, we have these hepatocytes. They are epithelial cells too, and we have the endothelial cells lining the sinuses, and there's Kupffer cells in there as well too. But again, those are epithelial cells lining blood vessels, are known as endothelium. Here in the hepatocyte, we can see lots of glycogen, and we see junctions between adjacent cells. There's a gap junction right here. The word gap junction, as we see there, means uniform thickness between two different cells. And the membranes of these two cells are uniform thickness because there's proteins along the way that have little channels in them for communication to occur between those cells. Also, if you look at this surface, this is the bile canaliculus. So the hepatocyte is producing bile, and it doesn't want the bile to go into the rest of the body, so it has a tight junction. There's a zonia occlutans, there's a desmosome right in through there, and you can see where the membranes are kind of fused together there to make a tight junction. These are intestinal absorptive cells, simple columnar epithelium, brush border, basement membrane there, we can't see that, but you see the connective tissue below that provides the nourishment to these cells. Here in the ileum, we see the brush border of intestinal absorptive cells, and we see the basement membrane, which is PAS positive as well. Here we not only see the intestinal absorptive cells, but we can see the ones with granules at the base. These are argentifin cells. Here we can see them here and here. Those are endocrine cells, but again, they are epithelial cells in nature. In the colon, we see intestinal absorptive cells with many goblet cells in the case of the colon. If we look at one of those cells, we can see this is one cell. Here's this nucleus, another cell here, another cell here. We can see the interdigitation between adjacent cells. We've already looked at the Golgi apparatus, coated vesicles, budding ER, and the various components that we can see. But here you can see there's no really junctions. We don't really have desmosomal connections that many, so that the cells can actually open up as they increase their secretions. Lining blood vessels, we have endothelial cells, simple squamous epithelial cells, which are endothelium. Endothelial cells line blood vessels, as we see here. Here's a little marginal fold which kind of forms over the junction to help prevent bombardment with red blood cells and white blood cells going through. Here we can see in a spermatic cord the lining cells of the various vessels, the arteries, the veins, or the capillaries, or the venules. All those have endothelial cells. Here in a vein we can see a valve, and in that valve we can see that they are lined by endothelial cells, simple squamous endothelial cells. This lymphatic has endothelial cells on the surface as well. But here we can see the endothelial cells on the surface of that vein. Electron micrograph that we see, this is a muscle cell, but here we see an endothelial cell. And you can see the junctions, the tight junction between adjacent endothelial cells, as well as a basal lamina. You can see the basal lamina through here, and then there's another tight junction between another tight junction, the zonia occludens, between two portions of cells lining this lumen. There's a lumen here, there's a grid bar there, artifact, but here we see the lumen endothelial cells, and then that's muscle cells of a little capillary. We've seen this slide before for the macrophage, but today we're supposed to look at the Sertoli cell. And this is a Sertoli cell from a real old rat. And you can see that the basal lamina is very thick. It's much more thicker than it should be for a routine epithelial cell. And this is what happens with advancing age. Sometimes you get the thickening of the basal lamina is so large. In the skin, we have stratified squamous epithelium. These are the epithelial cells that divide from here and differentiate as they move to the surface and become keratinized on the surface. And here we can see glands. These are sweat glands associated with dumping sweat on the surface of these cells. Here again, we can see this is in the scalp. This is the hair follicles. The hair follicle itself is continuous with the epithelium on the surface. Indeed, it is epithelium, and it's the source of the hair itself. So cells are transformed into the hair, and those cells are epithelial cells. Here we can see the finger pad, stratified squamous epithelium, non-vascularized, you see, gets its nutrients from the connective tissue below. Also in there we can see the sweat glands. So these are the sweat glands that dump their content on the surface to leave your fingerprints. Stratified squamous epithelium with the different layers, the germinal layer here, spinosum in through there, and then ultimately we have the stratum corneum at the very top. And right in through there you have stratum granulosum. So stratum basal, stratum spinosum, stratum granulosum, stratum corneum are the different layers that we would have. And we'll learn that when we talk about skin, but today we're just talking about that these are epithelial cells that have differentiated from down here and moved up as they differentiate, lose their nucleus, become granulated, and then ultimately sloughed off. If they are exposed to air, they become keratinized. If they're not exposed to air, like in the vagina, and they stay liquid or either in the oral cavity, it's non-keratinized stratified squamous epithelium. And so here we can see the sweat gland as we see it. It looks like it's just cut, but actually it's really a coiled gland with a duct that runs up to the surface. And so here we can see the sweat glands. The duct, it has a characteristic stratified cuboidal. They're kind of cube cells, more eosinophilic. You see it's red, red, red here, less red here. This is a secretory, 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 duct, 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 duct. And so the ducts going up to the surface is what we're seeing. Here we see a little venule, and these are the endothelial cells that line that. Of course, again, epithelium in nature, and these are epithelial cells as well. If we look at an electron micrograph of the skin, we would see the stratum corneum and the stratum granulosum. And this is a stratum spinosum, where you see these spines. These are actually the desmosome connections, and the dark here is the intermediate filaments. And it's the intermediate filaments that persist as the core of the cells up in the stratum corneum. And then the basal layer, you have cells that proliferate. Here's a melanocyte that produces pigmentation for the cells. Here we see some capping going on, but the point here is stratified squamous epithelium is a protective, resist mechanical stress type of epithelium. In contrast to the stomach, we have simple columnar epithelium. This is the surface of the stomach, and you can see the surface mucus goblets there, which facilitates a release, keeping them lubricated at the top so that the cells will not get digested during the stomach. In the urinary bladder, we have a transitional epithelium, and we can see them. You have kind of scallop type surface whenever the bladder is relaxed. So it's transitional epithelium in the urinary tract. Also in the ureter, again, we have transitional epithelium surrounded by connective tissue and muscle. In the penis, the urethra has varying types. It could be transitional, could be stratified cuboidal, or stratified columnar. Different types of epithelium lining that cavity. Here we can see this looks like a stratified cuboidal type of epithelium, but in other places it looked like it could be transitional epithelium, but they have both kinds. So you see that the body has epithelium throughout. It's got epithelium lining the skin. The epidermis is really epithelium. It's got epithelium in the oral cavity, in the esophagus, in the stomach. It's also epithelium makes glands, and different types. It all depends upon the function, whether it's ciliated to move mucus out, or where it's transitional so it can stretch as it need, or a simple columnar to aid in absorption. We have these different types of epithelium that facilitate, and their shapes and number of cells facilitate their function. Thank you.